Hello, my name is Kit Fuhrman. I am a GeoMx product manager for the Cancer Transcriptome Atlas. It's a pleasure to be here today to talk to you about DSP, the Digital Spatial Profiler, and the Cancer Transcriptome Atlas. So this is a great technology from NanoString, of course. As a company, we've been around for quite some time, and many people know us for gene expression platform, the Encounter analysis system. But in 2019, we actually launched a new system called the DSP, Digital Spatial Profiler, that does something really unique and delivers great spatial information along with both protein and gene expression information. So, and that's really what I'm going to focus on today. So I'm sure many users here perform some type of bulk gene expression. It's a great technology, whether it's on the Encounter or you're using RNA sequencing. And it tells us a lot about the biology. Here is an example of a signature that was actually developed by Merck. It's called the Tumor inflammation signature. It was developed on Encounter, but this concept is definitely extendable to other disease pathologies. And it basically tells the user whether or not the tumor itself is inflamed, whether or not there's an active immune system. But there's a lot of caveats to these signatures because, it does predict response to therapy in this case. But there's many patients where there might be a high score, but no response. And so we know that it's not a perfect measure of whether or not somebody will respond to a particular therapeutic because there's such a diversity in the biology. So to illustrate that with images, I have two colorectal carcinomas here. And these tissues have almost the same inflammation score. We can see there on the top right of each section. So two different patients. One has the immune system on the left in magenta. You can see that it hasn't really infiltrated the tumor, which is in green, which is pancytokeratin. But on the right, you can see this really highly infiltrated tumor where the immune cells, the CD3 T cells and the CD45 cells are infiltrating into the green portion. And so these two patients, even though they have the same level of inflammation, or same level and same scores, are gonna have two very different responses. And so, what's lost in these bulk gene expression assays is some of this spatial information. And that's why it's so important to help us understand the biology of cancer, but and generally biology across the board in all different domains. So that's really where the Digital Spatial Profiler comes in. It helps you take this high-plex gene expression information or protein expression information and merges it with the spatial data that you get from a tissue section, right? So you both get the precision and quantification that you would expect from these counting assays, but also kind of the contextual information of where this signal is actually coming from. So on the DSP system, it basically takes a standard tissue section slide. And so, there's numerous different sample types that you can use on our particular platform, both FFPE, which we've optimized all our assays for, so formalin-fixed, paraffin-embedded samples, or, fresh frozen samples. A lot of people will use tumor microarrays on the platform. So it's pretty flexible in the input. As long as you can put it on a slide, you can run it on our platform. So this is a nice general overview of the DSP process. On the left-hand side, you have our reagents. And so this is a lot of where the power is in the platform. We either use antibodies for protein detection or ISH probes for RNA detection. And so you have on each probe is attached a DNA oligo or DSP barcode. through a UV photocleavable linker, right? So this allows us to, stain these tissues en masse with the probes. And then what the DSP system does, it allows you to visualize your tissue, then select a region of interest, and then it shines a UV light in a specific region that you've selected. And it illuminates only that region while leaving everything else untouched. And it leaves your tissue intact, very importantly. It doesn't destroy your tissue. Because the DNA oligos, the DSP barcodes, are attached via UV photocleavable linker, when the UV light hits those specific regions, the DNA oligos come off the tissue, and then we collect and distribute them in a well of a 96-well plate. So then that allows those DSP barcodes or the DNA oligos to be counted on the back end. So what that gives you is a very distinct profile, quantitative profile of what's expressed in each region of interest. So you can see on the right, you have each circle there. This is a lymph node biopsy. And you can see each circle, you'll have a unique profile for every single one of these. So instead of having a gene expression signature across the whole tissue, you can now get a gene expression or protein expression profile for every single region that you've selected. And so now you can place those regions in spatial context. So just real briefly, we have a number of different reagents. So we have protein assays for immuno-oncology, immunology, as well as neuroscience panels. So this is really our protein offerings. We have pre-validated content for formalin-fixed paraffin-embedded samples. Right now, everything's enabled to be read out on the nCounter platform, meaning those DNA oligos that come off the tissue are read out on the nCounter, and we're building out the capability to have those read out on the NGS platform. Now, the readout of these NGS, DSP barcodes or DNA oligos, the readout on for our small plex panels for RNA are in the nCounter. And then we've expanded the readout using the NGS system, so Illumina's NGS readout, so that we can actually read out a large number of genes all at once. So for the Cancer Transcriptome Atlas, we have the ability to read out over 1,800 genes at one time from every single region of interest. So this is obviously a large amount of information from every single region. So instead of looking at the cancer transcriptome across the whole tissue, you can look at it at various regions throughout your tissue. And so these have the 1,800 genes here are specific to immuno-oncology. And then just to introduce you into what we're doing, we're launching next year, early next year, is the whole transcriptome atlas. And so this will be for human and for mouse. The other two panels were for human, but this will be you'll be able to measure basically the whole transcriptome in every single region of interest. So a truly unbiased assay. So all the protein coding genes. So to focus a little more on the atlas that we use today, this has over 1,800 genes. It covers a wide array of pathways. So everything from B cell and T cell biology to stromal architecture and the immune microenvironment. Also quite a bit of tumor biology. And so this targets things like cytokines and chemokines and growth factors and so forth. There's a lot of annotations in this particular panel. We've borrowed a lot from our nCounter panels, so it has a lot of the immuno-oncology signatures that you would expect, like PAM50 or the tumor inflammation signature, or a set of genes. And of course, there's the ability to add custom targets. So this is the panel that I'll show you some data with today. And this is actually read out on an Illumina sequencer. So once you have collected the barcodes, the DNA oligos from the tissue using the Digital Spatial Profiler, you'll simply read that out on the Illumina sequencer and then import that data back into our system. So just to review, you'll take your section and you'll stain it with both imaging reagents, which is an important part of the ROI selection strategy. These are fluorescently labeled antibodies, or they can be ISH probes. RNAscope is compatible with our platform. And so you'll stain it with these imaging reagents. And then you'll also use our profiling reagents. So those are either the protein panels or the RNA probes, the CTA in this case, the Cancer Transcriptome Atlas. You'll prepare the sample, and then you'll place it on the Digital Spatial Profiler. And on the instrument, you'll actually select your regions of interest. And I'll kind of dig into what that actually means and the flexibility of that process. But then the system will allow you to cleave the oligos from every specific region. It collects those oligos and dispenses them in a plate, in a well, in a 96 well plate. And then you count them on either the N counter or the Illumina sequencer. So this just shows you the reproducibility of the platform. These are on the right-hand side. You can see two serial sections, lymph node sections. And then you can see the reproducibility of the correlation between selecting the same region across two different serial sections. So 0.983 R squared value. This high level of reproducibility allows you to, basically measure biological change, very precisely. And you can see another thing to point out here is that there's a large dynamic range, as you would expect from this many targets. So you see things that are highly expressed, but you also are able to discern low expressors as well. So up to this point, I've shown you these spatial images with circles, basically regions of interest, where we've created a polygon or a circle, selected it, and then illuminated that particular circle. But on the instrument is a digital micromirror device that allows us to finely shape the UV light source like a mask. And so here in the middle, I have this is just a core from the TMA. And you could just create a circle and take the whole core as one profile. But using this DMD device, we can actually cut apart the tumor and the tumor microenvironment from one from another. So it actually allows us to finally dissect out the two different signatures. And so in this particular example, we'll get a profile for the tumor, which is on top, and then we'll get another profile for the tumor microenvironment. So we actually have two distinct either protein or gene expression profiles for what we're trying to study. And this really allows you to see the interaction between those two different segments. So we've been running projects for quite some time. And so over the years, we've developed different profiling strategies. So obviously the simplest approach would be geometric. And many people like to do this because it allows them to just place circles or squares or polygons over regions that have some morphologically distinct aspect to them. So, putting them in regions with heavy amount of infiltrate or positive for a particular signal or a particular morphological feature, such as a part of the anatomy. So they can simply move these circles on top of it or, shape a polygon to kind of fit whatever they're trying to study. You can take this a step further and segment. So basically segmentation allows you to basically segment on the fluorescence that you're looking at and say, I want everything that's positive for pan-cytokeratin, or I want everything that is negative for a particular fluorescent marker. And so it allows you kind of, like we said, cut apart different segments from maybe the tumor from the immune cells that are infiltrating it. And then just an extension of that would be looking at, cell type specific population. So basically selecting all the T cells or all the B cells that are in a particular region. And then there's also contouring, which allows you to look at biological gradients in a particular tissue. So going back to our example here, we have two very different tumors that have the same bulk signature, but we'd want to profile them so that we could really pick apart what's happening here in a spatial manner. So you could just draw geometric circles over each one of those. And we can see here at the top, we have non-infiltrated tumor regions, right? And so you can see that in green. And then at the bottom, we have these infiltrated tumor regions. And it's not annotated here, but the pink on the left are really the immune environment. And so the infiltrated tumor regions look a lot like the immune positive regions, right? The magenta, which is CD45. And so we can really tell that there's something unique about the tumor in both regions. One, in the top tumor, somehow the tumor is excluding the immune system, whereas the immune system in the bottom one is able to get past that barrier and infiltrate. So we really, we have this tumor immune boundary here. We really want to better characterize it. So how do we characterize this this invasive margin? And so one way to do that would be to select these 3 different regions of interest, and then we can actually contour across the region. So we draw the boundary, and then we're able to segment each one of these regions across boundaries. So we've taken 3 regions of interest, and we've basically split it up into different contours. So every contour here is going to have its own profile. And so that allows us to ask a lot of questions about the tumor immune boundary in this particular patient. And so we can see the gene expression profile change from when we go inside the tumor to outside. You can see NET gene heavily expressed within the tumor. We can see the HLA-DRA gene expressed in the tumor and then across the boundary. Of course, chemokines are not highly expressed within the tumor, but then MHC class II molecules are highly expressed along the border, but then decrease the farther away that you get. So you really get to see in this particular ROI the changes across that boundary and how the biology is, how the tumor is interacting with the immune environment. And then the great thing about the DSP platform is built for large cohorts of samples. So here we have 2 different patients. One is the highly infiltrated tumor, and the other one is the immune excluded tumor. But the system is really designed to have large cohorts, whether they're TMAs or you're running 24, 48 slides through the system. You can take all that data and put it together. So here we're looking at, the differences in the immune response of CD8s and CD3 T cells across the immune boundary. And each dot here is a different measure at a different point along the boundary. We can see things like, in this case, the inflammatory macrophages are enriched in the inflamed tumor with the heavily infiltrated T cells. We can see that distinct difference here. And, this kind of shows us that there's a strong M1 and M2 macrophage polarization with that particular patient that has heavily infiltrated immune cells into the tumor. And we can also take this data and we could, apply pathway analysis to it. So you can take things like, an interferon signaling pathway, and you can basically measure it across the border. And what we're seeing here is, we've taken many tumor stroma boundaries across the whole tissue, and each line here maybe it could be a different boundary. So there are other things that we can ask with these particular samples. Maybe we want to know how the T cell functions are different depending on if it's a infiltrated tumor versus one that has a strong boundary. And so we can do that. Here we've selected regions that are inside the tumor, and we've selected regions that are outside the tumor. So we want to know how the T cells are changing if they're able to get inside the tumor. How are they able to overcome this barrier? And why are, some T cells kind of hanging outside the tumor boundary? And so what we're able to do is basically select all the T cells, and then we can select everything that neighbors a T cell, right? So you get both this profile for what is happening in the T cells, and then you can see how it's actually affecting the environment around it. And so we can see things, obviously, just as a control, CD3 is highly expressed in T cells, obviously, and lowly expressed in the neighbor compartment. But then you have, things like TIM3, which is upregulated in T cells within the tumor, but not so much in the stromal environment when a T cell is in the stromal environment. Same with 41BB. So you kind of pick apart the different T cell profiles for those that are inside the tumor versus outside of the tumor. And this is just some follow-up experiments that we did with interferon gamma in RNAscope to show, that inflamed phenotype. And so the last thing I want to show you is just an example that that a customer did for a conference earlier this year. And so one thing that we found is a lot of people have either bulk or single-cell RNA-seq information that they want to integrate spatially. And so single-cell RNA-seq has been great because it tells you a lot about these populations of cells, but it doesn't really tell you where these cells are. And so we've developed methodologies to help our customers do just that. So this is Alex Warwick from the Garvan Institute, and he's been working with triple-negative breast cancer. And so he performed an analysis with actually our whole transcriptome Atlas assay, where he was looking at different cell types in the TLS structures within the triple-negative breast cancers. And so just to give you the high-level overview, he took his single-cell RNA-seq signatures because you can identify these very clear populations of cells. He then created ROIs here in the middle, and then segmented on pancytokeratin and then the stromal environment. So really everything else that it wasn't tumor. And then using these signatures, he was able to deconvolute the data and understand the composition of each of these segments. And so you could actually tell which cells were in each of these segments in these two regions and then across his tissues in general. So it's a really good way to kind of integrate both your single cell data, whether it's in cancer or neuro or any other tissue, and take your single cell data and then use it in your GeoMx DSP experiment. So with that, I want to thank everybody for your attention and interest. Feel free to reach out with any questions. You can find this single cell decon on bioRxives, the algorithm that we've just published, or more information about GeoMx and the Cancer Transcriptome Atlas by emailing geomx@nanostring.com. Thank you.